So what's happening 48 hours from now? Test, exactly. If anyone has accommodations and they haven't gotten in touch with me, letting me know how they are planning on doing the exam, please do so ASAP. And particularly if you're taking your exam in the testing center, I need to know to get that exam there. I may forget, and that's rather embarrassing for all concerned if that's the case. Second thing, probably hopefully most of you have noticed, one of your colleagues reminded me that I was going to post clicker questions. There's now a PDF with all the clicker questions that we've had so far posted on it with all of the answers that are on there. I have regular office hours today. I'm having some extra office hours tomorrow afternoon. If people have questions, need to chat with me, feel that requirement. Also, feel free to email. Again, I will try and post all of those email answers, not just directly back to you, but also on D2L. Yes, you need the big blue Scantrons for the exam. Once you have started it, you have as long as you need to finish. Shouldn't take more than about half hour, 45 minutes. But if you do need longer, we'll figure out a place that people are still working on it can go, probably just outside here to continue. Any more questions about the exam? 50 multiple choice questions, no drawing out base pairs. I know you feel terribly sorry about that. Okay, so as usual, although probably most of you haven't had too many of my classes before, today is just material that we've covered before. Things that I think I've done a bad job of explaining the first time around, things that more people have had questions about, and things that I think are potentially quite important. Whether these end up on the exam, I make no guarantees one way or another. So I'm probably going to return to this particular image quite a few times throughout the rest of the course. Basically, the sort of take-home message here, that I really didn't mention before, is that microbes rule. That's because there's far more molecular diversity in the microbes than there is in all other cellular life. And so I will beat on archaea quite a bit, but the main reason there is to think about the unifying features that are the same throughout molecular biology. And if you want to think about diversity, then anything that's present in all of these diverse organisms is likely to be really fundamental. And we're really concentrating on a lot of these fundamental things. Yes, sure, there are exceptions, chromatin, etc. Chromatin we're mostly talking about is going to be eukaryotic chromatin. But for the most part, they're really big generalizations. So that's the main thing that I wanted to mention here and repeat. And again, when we talk about archaea, it's mostly going to be my favorite organism right here, cellular organism anyway. So I've also mentioned conservation a number of times. This is getting back to that same sort of idea. Anything that's present in very diverse organisms is something that's likely to be really fundamental and important. And the reason for that is that over evolutionary time, these things have been conserved. And that's what conservation is really about. Evolutionarily, if something has changed here, it's bad, so you don't see it. And one of the important things that I like to remind people about as far as evolution is concerned is that evolution has gotten rid of all the things that don't work. It's not survival of the fittest, which is the ridiculous oversimplification. It's really what you don't see is what hasn't worked. So it's kind of a different way of thinking about evolution, but that's definitely the way I think about it. And that ties in really nicely to conservation. So in the region here in the red box, that's saying these are changes which have been maintained through evolution. And so they work, and they're probably not that critical. On the other hand, in the green box, there are very, very, very few changes that have happened. And so any change that had happened in here, because as we'll talk about after the midterm, who cares about after the midterm, do that first. No, but we'll talk about DNA being a really crummy molecule for saving your genetic information, because there's all kinds of damage and things that happen to it. So DNA damage and changes to nucleic acid happen all the time. And so the fact that if you look at extant organisms, particularly really diverse extant organisms, have the same sequence in all of these, means that a lot of things have died out because they haven't made it. So that, again, please stop me as I'm going through some of these things if there's anything that's not clear. Central dogma, probably beating this a little over the head. DNA goes to RNA, goes to protein. We did mention reverse transcriptase for making cDNA libraries. That's this transfer here going from RNA back to DNA. It's the reverse transcriptase enzyme that does this. Breaks the central dogma. Well, when Crick came up with the central dogma, he actually had arrows in all of these directions, including from translation back to RNA. But this is the major thing, again, we're going to be talking about. So far, we've just talked about the DNA structure, talk more about the RNA structure and transcription and replication. One thing that we have talked a little bit about so far are those changes that happen, which are the epigenetic changes, changes that happen in terms of changing nucleosome structure, et cetera. And that ties back to this environment issue over here, what genes get turned on and what genes get turned off at any given time. Homology, I think this is a nice example of where I messed up. Ortho versus para, orthologs and paralogs. So orthologs, straight, something which has a gene which is diverged due to speciation. So it's a vertical inheritance, and any orthologs are going to be two genes in different organisms that have descended by that speciation route. Paralog, I just like to think of this as the two parallel lines for those parallel genes, both present in one organism. So it's gene duplication that has happened, and then one, or actually usually both of those genes have happened to diverge after that point. So orthologs, two different organisms, paralogs, same organism. You got gene duplication in the same organism. Often those two genes can now undergo lots more changes. And that's really through both of these mechanisms that we end up with gene families. So again, we'll spend more time talking about gene families as we move along through the rest of the course. So this is, again, I think I did a really crummy job. In fact, switched them up in one of my lectures. So everybody clear on orthologs and paralogs? Yes, good. Okay. So again, this is not specific to, I'm going to ask like 8 questions on the exam on van der Waals interactions. No. The point here is to emphasize that most of the interactions we're going to be talking about, with the exception of some of the polymerization reactions, covalent changes, most of the interactions in biology are going to be weak interactions, non-covalent interactions, and lots of them. So the big advantages to that are that we have flexibility and dynamics. If it were really a very strong interaction, these things aren't going to be able to move around, and you aren't going to be able to change things. And so change is very, very important in terms of biology. So that's the main message that I wanted to have here. Entropy, someone was asking me about this also after one of the lectures having to do with what would happen if we let our kids do what they want to do, and then requires energy input to come back to something that looks like a state of order. And so this is really the life state, and you have to input energy. There has to be an energy input to go from disorder to order. And the classic example that we're using in this class, and particularly all of molecular biology, is going from nucleotides, mononucleotides, to DNA, any kind of monomer to a multimer. You've gone from disorder to order. Individual nucleotides floating around versus a macromolecule. Individual nucleotides are, at least as far as genetic information is concerned, worthless. So now we want to have them in some form that they can be used. And that requires an energy input. The energy input that's used for nucleic acid polymerization comes from the hydrolysis of ATP. This hydrolysis of ATP generates the high energy intermediate, which is true for many, many, many of these biological reactions. And when I'm talking about reactions, I really mean some kind of covalent bond, either formation or breaking of covalent bonds. At least as far as we're concerned, that's all the reactions that we're talking about. We'll talk about interactions. Those are all of those weak interactions in terms of van der Waals interactions, hydrogen bonds, etc. So here, the reaction is going to be first adding two phosphates from two different ATP molecules to your nucleotide triphosphate. And one of the things I think I did a really poor job of explaining before is this is the alpha phosphate, this is the beta phosphate, this is the gamma phosphate. And so what happens when you have the polymerization reaction, the DNA polymerase we're going to talk a lot about later, is that you have hydrolysis of this alpha-beta phosphodiester bond, releasing pyrophosphate, these two phosphate molecules together, which you have here. These get hydrolyzed to individual phosphates. What is this doing in terms of order versus disorder? You're going from phosphates connected to each other, phosphates not connected. So you're increasing disorder. So this is an energetically favorable reaction. You have to put energy in. And so this is what's helping to drive this reaction from monomers to polymers. Make sense? Yes? Everybody's happy? No, they're just too early in the morning. It's Monday. Spent all the weekend studying. Okay. So amino acids. I specifically mentioned 4 that I will expect you to remember. Those are conveniently the ones that are right up at the top here. Aspartic acid, glutamic acid, arginine, and lysine. I will try and always put these 3-letter codes, but we'll often talk about these 4. Last lecture, we spent quite a long time talking about K. Lysine 9 in the tail of one of the histones, which is getting modified either by methylation or acetylation. And important aspects about that lysine: positively charged, likes to interact with negatively charged DNA. Aspartic acid and glutamic acid, of course, are now negatively charged. They're not going to want to interact with DNA, at least not directly as far as the backbone is concerned. All of these amino acid side chains are usually going to be on the outside of proteins because they'll be interacting with water. And all of us, all biology as we know of it, is interacting with water. And that's partly because of the charge-charge interactions, but also because water forms really, really good hydrogen bonds with water. So those are our amino acids. We've talked a little bit about some of the other ones. Cysteine, this is the one which you can use to form disulfides with. A little bit in terms of serine, threonine, and tyrosine. These are the ones that have OHs and are getting phosphorylated by various different kinases. So we'll, again, talk more about those a little bit later on. Alpha helix, again, I think it's another case of where, oh, sorry, yeah.
I saw a question on the practice exam that asked what was like the charge of an overall peptide chain.
Yep.
So the way you answer that is you just look for which of the amino acids are positive or negative.
Yeah. So. Yeah, so the question was here, if someone were to give you a question on an exam where there was a peptide that had a sequence of amino acids on it and asked what the charge was at neutral pH, you would just go through and look for the lysines, the arginines, the glutamates, and the aspartates, and count up pluses and minuses. And I would try not to put a histidine in there because it's really confusing because it depends a lot more on exactly the pH that you have. Okay, other questions, things that have come up while you're looking. Okay, so alpha helix, again, I think I did a pretty crummy job of explaining this before. Main thing with alpha helices, and also true for beta sheets, is the way that these form is through hydrogen bonds that form in the peptide backbone. And so peptide backbone is basically everything except for where your amino acid side chains are, between these different amino acids at approximately 3.6 apart, but it's every single one because each of those is all wrapped around at about 3.5 excuse me, per turn. And what that means is every 7 amino acids, you're going to have the R group on the same side of the helix. So you got 1, 2, 3, 4, and we have 5, 6, 7. It seems to not be lined up here properly. Okay, 1, 2, 3, 4, 5, 6, 7. So these two are on the same side of the helix if they were properly drawn. This is a nice reminder that I did post an animation of this in 3D, which I find is a lot easier to look at on D2L. Another thing about alpha helices that I didn't talk about at the time, because we hadn't really talked about the DNA structure, is that it turns out, probably for really important evolutionary reasons, that alpha helices fit perfectly into major grooves of DNA. Surprise, surprise. And one of the great things about alpha helices, what does it do? It sticks the side chains out perpendicular to the axis of your alpha helix. And that means that those side chains can now interact with maybe DNA or other parts of the same protein in the interior of a protein, say what you have in the case of a leucine zipper, or interact with other proteins that could potentially be interacting with that particular alpha helix. So alpha helices, we'll see again and again, particularly when we talk about DNA interactions, but also protein-protein interactions, are very important secondary structures. Although secondary structures fold up together into tertiary structures. A couple of things about tertiary structures that I wanted to emphasize. One is that most amino acids are not involved in whatever the activity is of a particular protein. Most of the amino acids are just involved in making the structure of that protein. And there are lots of different ways to make structures of proteins. So you can have as few as 30% identical amino acids, and that's what these green ones are all supposed to be here, and still have an extremely similar three-dimensional structure. And those extremely similar three-dimensional structures will give you very similar functions. Here, elastase and chymotrypsin, both of them proteases, cleaving ever so slightly different substrates. And this is the kind of thing that you often see in protein families. So these could be orthologs of each other. They could be in different organisms or paralogs in the same organism. Structures, very, very similar, and having very, very similar kinds of activities. Now that we've talked a little bit more about the DNA structure, wanted to mention that the DNA molecule itself is actually relatively small compared to most proteins. And particularly when we talk more and more about proteins that are involved in both replication and in transcription, and of course for translation later on as well. Of course, when we've got RNA molecules, these guys are really big. And really big molecules interacting with DNA, particularly when that DNA is in a nucleosome, is clearly a problem. So how do cells deal with those problems? Modifications of nucleosomes, nucleosome remodeling complexes, etc. So just wanted to emphasize that particular case here. I think I did a crummy job of explaining the whole idea of non-structured proteins. So, didn't you just say, Dr. Stedman, that it's the structure which is leading to the function of all of these proteins, and most of the amino acids are not involved in any kind of activity. What's this whole deal with unstructured proteins? Well, unstructured proteins, mostly this is when they're under a particular condition. And often when we talked about the protein methods, we talked about X-ray crystallography and NMR spectroscopy for figuring out the structures of proteins. Well, if you don't have a protein that is crystallized, you're not going to get a structure for a protein that's not crystallized. So you don't know what the rest of that structure is. So it's actually a lot harder to figure out what these things are. So a lot of the unstructured proteins are proteins that we don't have structures for, as opposed to actually non-structured proteins. Most of these unstructured proteins will be structured in some way under a particular condition. And again, this has mostly to do with weak interactions. Classic example where we have binding of a otherwise structured protein to an otherwise non-structured protein. That's what this red one's supposed to be. This now has a very defined structure, the red one, but only when it's together with the green one. And so non-structured going to structured protein. You can have covalent modification that happens to an otherwise non-structured protein to give it a structure, here phosphorylation being one of them. And then some of these unstructured regions of proteins, I don't like to call these unstructured domains because the definition of a domain is that it has a defined structure, but if it doesn't have a defined structure, we shouldn't be calling it a domain. So a motif, or usually just a part of a protein, which is unstructured, but it's still providing some kind of function to some extent to your protein, or that rest of the part of the protein, because it's holding something that needs to be near to everything else in the protein. And so that's the whole idea of tethering, which is here. And then there are certainly proteins which stay disordered, i.e. not with a standard structure most of the time. And those are those that are forming things like the nuclear pore complex, but also we're finding more and more that proteins are going to be switching between two different kinds of phases. So phase separation, particularly in the nucleus, where you have sub-regions in the nucleus. People don't call them organelles because there's no membrane which is bound to them, but there's still a very clear region. And a lot of that actually has to do with the unstructured part of proteins, usually then hydrophobic parts, which are then interacting in sort of the oil and water way. So that you have, yes, it's mostly an aqueous environment in our cells, but there are some regions which are very hydrophobic. This would be one example of them here, say in the nuclear pore complex, but other ones as well. Sort of the classic example of different structures with different functions are the amyloid proteins, the bad amyloid proteins that I skimmed through really quickly at the end of one of the lectures. These are the ones which will precipitate when they're in an incorrect form and will be soluble when they're not. These precipitations are closely connected to neurodegenerative diseases, things like Creutzfeldt-Jakob, mad cow disease, but also Alzheimer's and Parkinson's. So these are normal molecules that are soluble, but they can also have a different form and be insoluble and probably contribute to disease. And how much they contribute to disease is a really kind of open question here. People always used to think these, amyloid proteins forming these different structures were always bad. Turns out that there actually are a number of good forms of these. Backing up a little bit, what happens with a lot of these proteins, like the tau protein in Alzheimer's disease, is they form these structures with two beta strands, and then these two beta strands all stack up on top of each other. And that's what seems to form these filaments, seems to form the precipitating proteins, and again is associated with disease. On the other hand, it turns out that there are some of these proteins that form these beta strands, yet also will be very important for serving as usually, again, one of these cases where you have like phase separation, where you've got lots of hydrophobic amino acids that come together, that then will be important for some kind of function. And when we talk more about where RNA sits inside the nucleus, we'll talk more about this as well. So all of these amyloids, double beta strands forming with each other can be both good and bad. The last sort of set of protein structure things that we've talked about so far have to do with modifications to structures that are used for signaling processes. First of these are the kinases and phosphatases. I like to really think of these as switches because once you have made a covalent modification, such as phosphorylation here, that's pretty stable. Now, have a signal that comes in, the protein kinase puts a phosphate onto your protein, and then there's some kind of structural change. This is a stable structural change. It's not going to come off until you have the activity of a protein phosphatase. So to the flip side of that are the G proteins, where you have a signal that comes into a G protein, usually bound to GDP, just two phosphates here. That will now bound to GTP, usually but not always in the presence of a guanosine nucleotide exchange factor. When you have GTP bound form, this has changed your structure, usually will be activated in some way. But this phosphate can be cleaved spontaneously. In fact, the G protein, GTP binding protein, are also known as GTPases. Because what do? They cleave GTP. And they cleave between the gamma and beta phosphate. So once this is cleaved, you're back in this state. This can be sped up by a GAP, or guanosine nucleotide activating protein. So yes, it can stimulate that activity, but it turns off by itself. And so this I like to think of much more as a timer. So it gets turned on by adding GTP, but then it turns itself back off. And so it's a much shorter time period where these are on and off. Okay, questions about protein structure before we start talking about protein methods? Okay, so protein methods, and actually this is a much more general method. It's not just proteins, but also true for nucleic acids. Gel electrophoresis, you put your favorite macromolecule in an electric field, and it will migrate from one electrode to another. If you have something which is negative, you want to have it migrate from a negative cathode to a positive anode, and you need something to separate these different macromolecules. And so this is the gel in this case, agarose gel, acrylamide gel, just has holes in it of different sizes depending on how much concentration you use of these different things, will slow down the way that these things are going to migrate through the gel. Bigger things with low charge are going to go very slowly. Smaller things with high charge are going to go fast. This is what happens in the case of what we call a native gel electrophoresis. So you're going to be separating based on size and charge. That is confusing for most protein analyses. So if you put in detergent, that will change this whole process, and particularly if you have a charged detergent like SDS, sodium dodecyl sulfate. So sulfate is a charge. The dodecyl group is going to serve as a detergent. That will unfold your protein, so it's going to normalize size and also normalize charge. So when you do SDS page, you're just separating based on mass. You're not separating based on the size and shape of your molecule as well. When we look at DNA, DNA is really nice because it's already universally negatively charged. So you can put DNA into one of these electric fields and it will migrate based on its size until it gets really, really big. And then we have pulsed field gel electrophoresis processes. So this is great for usually analytical purposes. You can't put very much onto one of these gels unless you make a really, really big one, and it starts to get very confusing. So usually for purification purposes, we like to use columns. And again, I should have brought one of these, and I'll just pretend that this is a little column here. So they're cylindrical, and what you do is you literally flow liquid from the top to the bottom. That liquid contains what you want to purify, but also a whole mixture of other things. And then in the middle here, you have something which slows down all of the different things that you have in your mixture, and hopefully will separate what you want from everything else. Now, there are multiple different ways that this happens. different kinds of things that will slow them down, but I did also wanted to mention this whole process. And so as you're flowing things through here, there'll be drips that are coming out of the bottom of your column here. So what will happen is the first 5 or 10 of these are going to have nothing in them. And then 10 to 15, nothing. 15 to 20, now what is this red stuff? 20 to 25 will have the green stuff in them. And so you're literally taking this mixture, you got the black at the top, and separating it into these different tubes, which will have different things in them depending on what's in your individual matrix. So what you need to do then is each of these individual fractions, if you're lucky, they've got these colors, and so you can say, hey, I'm looking for the red one, and I'm looking for the green one. That's very, very rarely the case. And so what you have to do with each of these now individual fractions, you've taken the stuff at the top, you've split it into some very small pieces. Now you need to go through each of those and figure out what the one it is that you're looking for. So we had a bunch of slides that had blue peaks on them in this lecture. So that blue was whatever you were looking for. Could have been an enzymatic activity, could have been a DNA binding protein, could have been a band of a particular mass that you see on an SDS page gel, etc. So just doing the chromatography doesn't really tell you what you have and which of the fractions you have it in. You have to do some kind of test to figure out where your favorite protein is in each of these samples. This is made a lot easier if you have some specific tag that you can have in your matrix here. So this would be our affinity chromatography. One of the main ways that people do this is they add an extra piece of protein coding DNA to their favorite gene so that their favorite gene will now have a tag that's associated with it. This will be a tag that binds specifically to nickel. In the case of 6-histidines, it could be a protein that binds specifically to glutathione, glutathione S transferase. One of the ones we use is what's called a maltose binding protein. And these are all added separately through recombinant DNA techniques, PCR or straight restriction endonucleases, this red piece right here. So this red piece is now attached to your protein because we know the genetic code. We can add this extra piece of the protein. This will encode a piece of protein. It's supposed to be dark green here, unfortunately a little hard to see. But that dark green piece now can be used for affinity purification, but it can also be used for localization inside a cell, for instance. So you have your favorite protein. You want to know if it is located in the nucleus. So you have a tag. You put it onto your protein, you put it into the cell, and then you look and see where the tag is. And since that tag is attached by peptide bonds through translation to your protein, it will tell you where it is inside the cell. So this is probably way 90+% of proteins are purified these days, if not 95, is just by adding a tag to them, because it's so much easier than going through this whole process. But if you don't have a tag, you don't have a gene, then you have to go through these other ion exchange chromatography, hydrophobic interaction chromatography, etc., in order to get to this point. And we'll talk more about DNA binding proteins later on in the class. Western blots, southern blots, northern blots, southwestern blots, northwestern blots, far western blots, etc. The whole idea of these blots is that you have transferred what you've separated onto a gel onto a membrane for really two reasons. The first one is to concentrate whatever you had. Usually these gels are relatively thick. Some of the agarose gels can be a centimeter thick. Acrylamide gels usually a couple of millimeters thick. Now what you're looking for is on a surface. And that surface is now something that you can use tools to try and detect things with. So the classic example in terms of protein detection are the Western blots. So Western blot, you transfer your protein, indicated by all of these bands here, to a membrane. And then you have an antibody that's going to recognize one and only one of these particular proteins because it's a specific antibody to your protein. This then has some kind of label attached to it so you can detect it. So here is now your Western blot. Your antibody has just interacted with that one particular target protein. You can also have DNA hybridization, which you use to detect a particular DNA. Works in exactly the same way. Now, instead of having an antibody that's associated with this band, now you'll have a complementary DNA sequence, which has some kind of label on it, which will bind to and identify this sequence. So the Western blots used for proteins for a second. Southern blots are going to be used for DNA. Northern blots you're going to be using for RNA. Yeah?
You just answered it.
Oh, okay. Okay, questions on protein techniques. Now we're going to shift gears and talk about DNA. Yeah?
This becomes like a southwestern blot when you're using DNA to target your protein?
Yeah, so if you have a DNA that you're using to target your protein, actually you can do it both ways around. So you can use DNA to detect your protein, which would be the hybridization that way, or you can have DNA and use a protein to detect that DNA. Either way would work. Okay, other questions on these protein techniques? Okay, so the genetic material, DNA is the genetic material, can't be the genetic material because it's too dull and boring because we've only got 4 nucleotides. Proteins makes much more sense in terms of what you'll use for the genetic material. Well, these, bacteria, again, going back to that whole slide, very first slide that we talked about, diversity of life, right? Who cares about bacteria? They're not closely related to us. We only care about them if they're making us sick. Well, turns out that they have the same genetic material as we do, and they're a heck of a lot easier to work with. So the transforming principle that, again, nobody cared about at the time, had to do with the process of transforming these rough bacteria to these smooth bacteria. And actually, it's a rough colony of bacteria. Each of these colonies has about 1 million bacteria in it, to a smooth colony of bacteria, because these bacteria are producing a capsule, and that capsule protein, which is capsule protein which leads to production of polysaccharides, is what causes the mice to get sick. Now, it's way easier to look at these colonies on a plate than killing off all of your mice. And all that they did was take these guys and extract everything that they could from them and try and transform these colonies to those that looked like these. Then, once they had this material purified from these guys, then they tried to get rid of it. DNase killed the activity, RNase and protease didn't. And again, so there's a process of elimination, not unlike the whole idea with evolution. The things that are, that work are present, and things that don't work have gotten removed, and so the things that don't work are going to be the ones which are important. So DNase in this case. We now know how this process actually works. Now this was much later, and that is that the capsule gene, which is part of the chromosome in these smooth colony forming cells, that gene is actually released when you heat up these cells. That gene is picked up by these non-pathogenic cells. And this is a process called transformation. It's the transforming principle, but also it's a particular kind of bacteria that can pick up DNA from the environment and incorporate it into its own genome. And these are cells that are called naturally competent cells. Most cells can't do this. No, your cells can't pick up DNA from the environment and then incorporate that DNA into your own genome. It's probably a good thing, because if you think about all of the bacterial genomes that are in our guts, you probably don't want to be incorporating all of those DNAs into your own DNA. So it's a particular mechanism that only works in this particular case. So the transformation process. Part of the reason that nobody cared was because there are these boring bacteria, and maybe the time just wasn't quite right for it. Hershey and Chase probably had a better PR department as far as their experiments were concerned. Also, they had a simpler system to work with. They weren't killing off all the mice. They had also the advantage of the development of the electron microscope, where you could actually see what was happening in this case, as opposed to where they could. So going back here, this bottom part is only stuff that was figured out much later. All that Avery and his coworkers could see was just a transformation between these different colony types. Who cares about transformation of different colony types? But viruses are so cool, you got to use those. Now, there was a whole group that was very interested in viruses, and in fact, a lot of these were people who were working on the Manhattan Project during the Second World War, who then decided, biology was more interesting than physics, and so they would start working on these kinds of systems. And that was the whole group that Al Hershey was involved with. So, Hershey-Chase experiment, of course, is you could differentially label bacterial viruses either with S35 that labeled proteins, P32 that mostly labeled DNA, and do an infection process, remove the empty shells, which you could only see that you'd done this using electron microscopy. And this is the famous blender experiment, the E. coli milkshakes, where you have removed this, and then you look and see what is maintained genetically through the next generation. It was the P32. Since P32 is mostly DNA, then you say P32 is the label for your genetic material. So let's move on to Crick and Watson. Wanted to emphasize this process here. A couple of people in office hours were asking me about it. What did they know even before they came up with their model, which I brought my own one here. Anyone wants to come up and check out everything that's wrong with it, please feel free to come and chat with me about it. There are a couple of major things and lots of minor things that are problematic with it. But just to walk through this process, what was known is that DNA was made up of 2' deoxyribose. So 2', what we need to do is talk about these labeling in the actual sugar that's part of DNA. So here's our ribose, 1, 2, 3, 4, 5 carbons. Each of those are primes because the individual numbers, 1, 2, 3, 4, 5, 6, 7, 8, 9, or 1, 2, 3, 4, 5, 6, are already taken by the base. So when you talk about modifications to bases, those are the single number. The prime numbers are now going to be what's happening in your sugar. So 1', that's where the base is attached. That was known. 2', deoxy, an H instead of an OH. 3', OH, this is where the extension takes place. This 3' carbon, which has an OH is attached to it, is now going to attach to phosphate attached to the 5' carbon with an OH on it. So every time I'm talking about molecular biology polarity, 5' to 3'. If you are confused about 5' and 3', come take a look at the model. See where your 5' and 3' are. And so you have an individual nucleotide. This is your monophosphate. It'll be a triphosphate, again, through those 2 ATPs, and then be incorporated into DNA. What did Watson and Crick do with their model? First thing they found was the only way they could get their bases to interact with each other is if the polarity was opposite. So one end is 3', the opposite one is 5'. And this always, again, it's going to be a 5' end. Here's your 5' attached to the 3' of the next ribose, 5' of this ribose, 3' of the next ribose. Over here, 5' is attached to 3', 5' is attached to 3', etc. This process of having anti-parallel allowed them to make nice, even base pairs across the structure. And very importantly, and something I didn't think I commented on well enough here, is this bond between the 1' position of the ribose and the base. Where this bond is, right here, also called the glycosidic bond, that's in the same place where you have a TA base pair, a GC base pair, an AT base pair, or CG base pair. This glycosidic bond is always in exactly the same place. So you could replace everything in the middle here and still have the outside be exactly the same. And that's the real key to having this be useful for genetic information. There's no change in the overall structure, and you can have any kind of information there. You can have any different kind of sequence. It still fits into the double-stranded DNA molecule. Another thing that I wanted to point out here, just as you're looking at the way they're put here, of course, they're wrapped around each other. It's the double helix. But here you can pretty clearly see we've got a major groove at the top here, minor groove here down at the bottom. Major groove, easiest way to see that, where are your methyl groups? And if you're confused about this, come take a look at the model up here. Where's the methyl group? And this is the side of the base pair that gives you, through chemical interactions, tells you what's there. The other side, you can't tell which ones of these are which relative to just the chemical groups that are in this side. Yes, in the back.
What is everything that Watson and Crick got wrong?
What is everything that Watson and Crick got wrong? Well, Watson, well, don't get me started on Watson. But in their actual study, the main thing that they got wrong specifically in their original model was the 2 hydrogen bonds versus the 3 hydrogen bonds. And again, the thing is, in the model, everything works perfectly. But it's not the way DNA looks inside a cell. It's, again, as we mentioned before, if you take the DNA from one of your cells and stretch it out in B-form DNA, it'd be about 2 meters long. Well, it's all packed into a couple of microns, like 10 microns inside a cell. So there's no way it could be like this from that point of view. But their model, fitting Rosalind Franklin's data, was really pretty good, again, with that exception of the different hydrogen bonds. Okay, more questions on DNA structure. Again, this is a great model for looking at. Everyone should have one. So one of the things, again, with this base pairing interaction, base pairs are very, very specific, and the only way you can make this nice double-stranded form is if the A's are matching with the T's and the G's are matching with the C's. And what this means is if you take 2 strands of DNA and heat them up so they come apart, and then cool them back down, as long as you do it relatively slowly, they will find their match. And they'll find their match extremely precisely. In fact, even in the whole human genome, if you've got 6 billion base pairs, well, lots of repeats too, so it's a bit of an issue, but you have a unique sequence, you can literally take the whole human genome, denature it by heating it up or other kinds of chemical purposes, and then put in a specific small piece of DNA that has unique sequence on it. It will find that one place in the human genome and bind to it. And that's, if we get to those karyotype slides, we'll take a look at it in just a second here. So what I'm trying to mention here is that this process is, heating up, the 2 strands come apart. It happens very rapidly in a relatively narrow temperature range, and the melting temperature is halfway through that transition. The way that this is measured is looking at the absorbance at 260 nanometers. Single-stranded DNA absorbs more than double-stranded DNA does under those conditions. But this is a completely reversible process. You can take the 2 strands apart by heating them up. They'll come back together properly. You heat them up, you cool them down. You heat them up, you cool them down. This is how PCR works, is that you pull the 2 strands apart, you let them come back together, you pull the 2 strands apart, you let them come back together. This hybridization process is not only dependent on temperature. A couple people asked me about this. You can put in other chemicals which will help pull the 2 strands apart and have them come back together. Also, the salt concentration that you have mixed into your solution will change the temperature that this happens. But still, the whole idea is you heat up your DNA, it will come apart. You cool it down, it will come back together. The actual number for the melting temperature, which is that transition again between all double-stranded, and all single-stranded will vary depending on what other kinds of chemicals you have in there. And so that's what's supposed to be shown here, where you have single-stranded DNA probes labeled in some way. We talked about radioactive labeling. We talked about non-radioactive labeling. Here's single strands. Here's another mixture of single-stranded DNA molecules. The only one which is complementary, these should actually all probably have primes next to them, is this A DNA. This A DNA will match perfectly with these probes at higher temperatures. At lower temperatures, it can match with some of these other DNAs. So sometimes you're going to want to find similar sequences to the sequence that you have in your probe. Sometimes you're going to want to try and find an exact match to the DNA that you have in your probe. Really depends on what you're trying to do in these particular experiments. DNA libraries. Why should we play around with DNA libraries? Couple of reasons. One that we really haven't talked about yet, we're going to spend a lot more time talking about later on, is when you have genes, these genes usually have relatively short exons, which are protein coding sequences. That's these red ones right here, and lots of green intron sequences. That's partly why our DNA is 2 meters long for each of our individual cells. Lots and lots of that is non-coding sequence or non-protein coding sequence. So very often you're interested in what protein coding sequences are actually being transcribed, made into RNA in any particular cell, any particular organism at any particular time. Kind of a snapshot of what the RNA is that's being made in a particular condition. To get that snapshot, you can make a cDNA library, and all the library is just all of the RNAs that are being made at that particular time now put into various plasmids, and all of those plasmids separated. Again, you can literally buy tubes of these from all kinds of different genetic suppliers. So here, all of these RNAs have been transcribed from the DNA. The introns have been removed. You've had poly A tails that have been added. Using reverse transcriptase, you can make copies of each of these RNAs. A couple things about this. A, of course, you're missing all of these introns, but also any RNA that more is made of, you're going to have more copies of the RNA. You're going to have more copies of the DNA. So you'll have 10 copies of this textbook and only 1 copy of this textbook. And this, of course, is the one that you want. But this process here gives you that snapshot. If you want to have a random distribution of your whole genome, say you want to sequence the whole genome, you don't really care what proteins are being made at any given time, then you want to chop your DNA into random pieces and then have clones of each of them. You might want to sequence all of these and then try and determine what the genome sequence is of all of these once they've been determined. Questions on these libraries? There seemed to be some confusion about libraries before. All right. Another thing that seemed to be a little confusing was this slide. You know, what are we talking about in terms of protein expression? This is basically going beyond that whole idea of tagging. So adding a tag we talked about before. You have your favorite piece of DNA that you've added a little sequence to at the end, which is going to allow you to purify it or localize it inside of a cell. Here we also have a promoter sequence, which is going to cause transcription to happen. You have a plasmid, also known as an expression vector. You cut it with your favorite restriction endonuclease. You cut your protein coding sequences of restriction endonuclease. You put the two of them together. You put them into cells. Usually this will be E. coli cells. And then you tell the E. coli to make lots and lots of copies of this RNA, which will then get made into protein. The way that this happens is you know how this promoter is regulated. In this case, it's regulated based on temperature. A low temperature, you're not knocking the RNA codes for this protein. high temperature you are. In this case, it's a DNA helicase. And that is telling you, yes, you're getting good expression of your favorite protein. Now you can go ahead and purify this protein and figure out actually that it does have this DNA helicase activity. But this is taking advantage of recombinant DNA technology in order to make lots and lots of your favorite protein and usually then do biochemistry on it. That particular protein you have to purify away from all of the other proteins. So this SDS page gel has lots of other proteins that are in it. Each of these bands represents a different protein which is present here. So you have to have some way of purifying it. Hopefully, if you have a tag, then you can use tag for affinity purification. Otherwise, you can go through the whole rest of the process of protein purification. Yeah, back. Oh, so what kinds of restriction endonucleases would you use for this particular step? It really depends on what your sequence is that you're trying to put in there, and also what's the sequences that are present in the plasmids that you're trying to use. So most of the time you can actually buy plasmids that will have the exact sequence that you want in them, and then your favorite protein, your favorite gene, you can either find the right sequence to put at the end of it, or usually what happens is you do PCR, which is on the next slide, and add those restriction enzymes to the end of it. And that's usually the way that this is done. You can do blunt ligations. Anybody tried to do a blunt ligation here in this class? Think yourselves lucky. It's a pain in the posterior. So the problem with those blunt ligations is that they can ligate to anything, and the main problem is they just go back together. You end up with the same thing that ends up back together. So yeah, they're a drag. Doable, but a drag. Okay, so PCR. This is how you would add those extra little restriction endonucleases to the end of your favorite piece of DNA. The way that you can do that is instead of having here our primers, the reds and the green ones here, at the 5' ends of these, you add a little piece of DNA, and just literally a sequence of DNA that, for instance, would have the restriction endonuclease cutting site that you're interested in. That particular end piece of DNA will get copied as you go through the PCR process. But let's back up. So PCR, you start with usually a genomic DNA. Say DNA that you've isolated from Sulfolobus or your favorite virus or humans. So it's a really, really big piece of DNA. You heat that up because you can do that. Talk about hybridization. You heat them up, the two strands come apart, and then you cool it down a little bit, and you cool it down to slightly below the TM temperature of these specific sequences that you've just ordered, the primers for DNA replication. And the TM you figured out by the number of matches that you have between your primer sequence and the sequence that you're trying to amplify. Then you put in DNA polymerase, all of the deoxyribonucleotide triphosphates, and usually a thermophilic DNA polymerase because you just heated all of this stuff up, and then let that polymerase extend. The polymerase will go until it falls off. So it just keeps going here, keeps going here, keeps going here, and eventually will fall off. That's usually beyond where your other primer on the other strand has also extended. So you have these pieces now, your gray piece that you started with, the green and red ones, which are the pieces which were made. All of these are longer than the DNA between, in this light gray section, that you actually wanted to amplify. This is what happens after your first heating, annealing, extending. You do this next step where you heat, anneal, and extend. Now you'll have some of the primers binding to your newly made DNA, the green and the red here. The polymerase will extend on these until it gets to the end, because that's where the end was made from your primer, and it will stop right here. But you'll notice one of these strands is longer than the other one, and that's because up here they had no reason that they should stop. It's only after you take these guys, heat them up, and reamplify. In the third round of PCR, you end up with double-stranded DNA. That's what you're looking for at the right length. As you continue this process, heating up, annealing, extending, the ones that you want of the right length are going to expand exponentially. So you end up with literally billions of copies of these. Whereas all of these other pieces that you actually don't want, because they're harder to work with, these are going to only expand geometrically. So ends up with many, many copies of the DNA that you want. And if you're smart enough to add a little extra piece at the end of your primer right here, that will get copied. And that can be that restriction endonuclease that you wanted to add to your favorite gene in order to be able to clone it into your expression vector later on. Okay, PCR, this is only one of the things you can do with PCR, but it's a really, really useful tool. Kind of revolutionized DNA diagnostic technologies, etc. Wanted to mention shotgun sequencing because I think I did a pretty crummy job of doing this earlier on. The whole idea of shotgun sequencing is you start out with multiple copies of whatever piece of DNA you want to sequence. You randomly cut up each of those copies. And so this piece was cut here and here. This piece is cut here. This piece is cut here and here. These are all identical to each other, all of these different sequences. So then you sequence the ends of each of these DNA fragments. You sequence this end, you sequence that end, you sequence this end, all at the same time. You end up with all this sets of C's, A's, T's, and G's. And then you throw those into a computer, and what it does is it finds the ends that overlap with each other. So this sequence right here, GTT CAG, is where it started from this very end. This sequence finishes up GTT-CA. Well, GTT-CA is identical between these two. That means that these two sequences belong next to each other. And so you can hook this piece of DNA to that piece of DNA, which is giving you a longer piece of DNA. The reason this is so important is that our sequencing technologies, the original sequencing technologies that were used for the Human Genome Project, dideoxy sequencing will give you, if you're lucky, 1,000 bases. If you are using some of the newer technologies, the high throughput sequencing, those will give you hundreds of base pairs. Well, hundreds of base pairs with the whole human genome, you really have to have good ways of matching and putting all of these individual ends together. And so that's the reason I wanted to recover this shotgun sequencing, and this is what happens in terms of getting the whole human genome. And I tried again to get this picture. For some reason it works just fine on my computer. As soon as I try and present it, it doesn't show up. So let's not bother with it. Finish up. Chromatin, it's where Watson and Crick got it wrong. Well, Watson and Crick had a good model for Rosalind Franklin's data, but inside of cells, and what I really wanted to mention here is that this is not just the case for eukaryotic cells. Bacteria and archaea have exactly the same problem. DNA is too darn big. It needs to be compacted, and it needs to be accessible to all those big proteins, which I showed you in those comparison slides, relative to the size of the DNA. So how do we get that? Nucleosome remodeling, the chromatin remodeling complexes, nucleosome remodeling complexes, also chromosome, as they say, nucleosome breathing. And then the last thing that we talked about, and the last thing that will be on the exam, is modifications that happen to the tails of your histones. These happen in the N-terminal, but also the C-terminal domains. These are usually hanging way outside the nucleosome structure. These get modified. In some cases, it's modifications which will be pushing away the charge here, but usually the modifications are going to be to something which is going to interact with another protein. Those other proteins are histone code readers. Good luck on Wednesday. Come find me in office hours. send me emails. See you then.